Afternoon talks are largely about biology. The morning was astrophysics and cosmology. Before I say anything else, tomorrow I just want to remind people there are poster sessions tomorrow, a poster session tomorrow, and many of the posters actually are closely related to talks that are being given today. So I want to encourage you to go to that if you can. Our first speaker this afternoon is Daniel Kuebler, a professor of biology at Franciscan University of Steubenville. He teaches courses in evolutionary biology, cell biology, and human physiology. His laboratory research involves two major projects: understanding the relationship between metabolism and seizure disorders, and examining the effects that various biologics have on human mesenchymal stem cells. He's the author of The Evolution Controversy, a survey of competing theories, a very good book. I strongly recommend this book to you. That book critically examines various evolutionary controversies. He has published a variety of popular articles on science, culture, and religion. He received his PhD in molecular and cell biology from the University of California at Berkeley. And so he will talk to us today about the origin of evolution, the interplay of order and contingency.
Okay, well, thank you, Steve, and thank you to the organizers of the conference. This is great. I'm really excited to be here and to talk a little bit about the sort of next step up from cosmology as we look at evolutionary history of the evolution of life. And this talk is going to sort of focus on the answer. How do you approach answering two questions? One would be, why does biological evolution work at all? Why is natural selection a viable mechanism? All right? And two, why do we have this specific evolutionary history that we do have? So if you look at over the last 4 billion years, why do you have a specific evolutionary trajectory that we do have? And it looks at the interplay of two major forces, order and contingency, right? And if you ask people to walk down the street and ask somebody to describe what do you think of when you think of evolution, most people associate it with contingency. Say it's just a bunch of random things that happen. There's random mutations, random events, and so forth. You seldom get somebody that will say, well, it's an order. It's predictable. There's a lot of regularities there. And I think what I would like to sort of convince you today is that if you look at evolution, that the order in evolution is fundamental and is actually more important. The contingency in a sense secondary, although it is critical and a major factor in evolution. Okay. So the talk is three short parts. The first is just going over what I mean by order and contingency in evolution. The second is to talk about what structure or order is required for evolution to operate. And this is, I'll be very brief on this part because a lot of it was in a sense hinted at or covered in some of the cosmology lectures this morning. And then the last part is the question, does this order, the order that's there, significantly shape evolutionary trajectories? And what I'm going to focus on is protein evolution and see, is there order in the physics and the chemistry that sort of dictate or funnel outcomes in terms of what proteins evolve and what types of proteins evolve? Okay. So interestingly, if you look at the Origin of Species, and if you read Origin of Species, what you come away with from that book in terms of the interplay of contingency and order is you see a dynamic balance. You won't read that book and say, wow, it's all a bunch of random changes that go on. Okay, you get impressed by there's quite a few passages and quotes in the book that really stress the law-like order, predictability of evolution and of natural selection. I'll just sort of read the second quote that I have up there. And he says, Darwin points out that a grand, almost untrodden field of inquiry would be opened on the causes and laws of variation, on correlation, on the effects of use and disuse, on the direct action of external conditions, and so forth. So he talks repeatedly in Origin of Species about sort of the laws governing variation, right? And again, he didn't quite understand mutations and how they actually occur, but he thought there were certain patterns that you could expect. Laws of correlation. If one trait changed, another is going to change in a sort of predictable manner. It's not just going to be sort of random fluctuations. This is not to say that Darwin didn't point out the contingent events. He certainly saw that evolutionary trajectories are influenced by many contingencies as well. But there is a balance that goes on there in Origin of Species. Okay. That said, if you, in sort of popular culture, what you see is an imbalance. And that I think that what you see when you talk about evolution is that contingency dominates. And there seems that there's order might be mentioned in passing, but evolution is dominated by these contingent events. Chance encounters a meteor hitting Earth, and if that never happened, X, Y, and Z would have never followed, and so forth, rather than stressing and looking at the underlying order. And in fact, this is used often for arguments, about purpose and design in evolution. Okay, so the arguments will go something like this: evolution via natural selection is primarily a contingent process, mostly random chance events. And because there's high levels of contingency, this demonstrates, lack of purpose or plan in evolution, right? So you got these sort of 2 points. I'm not going to address the second point. I think both points are, in my opinion, are wrong, right? The second one, you can make philosophical arguments to point out the problems with that. But I want to look at the first one. Is evolution via natural selection primarily a contingent event? Okay. Certainly there are contingencies there, but as I mentioned at the beginning, I really think the order is what is fundamental. Okay? Now, when you are talking about order and contingency in evolution, the level at which you're looking at an evolutionary pathway matters. Okay? So there is, on the right, I have a diagram just sort of illustrating some experiments, the Linsky experiments. So these are long-term evolutionary experiments. So he takes, with E. coli, you allow them to evolve in parallel, like take 12 different strains and allow them to evolve for tens of thousands of generations. And if you zoom in on any, and so this diagram, each one of these green lines would be one of these populations, for example. If you zoom in and look at one of the pathways here, you're going to see pathways that are very contingent. You're not going to repeat these. If you ran it again, you're not going to see the exact same set of mutations and changes and variations going on. Okay? But if you back up and look at the whole picture, if you zoom out and look at the whole thing, what you see is many of these pathways end up at very similar places. Okay? They don't all end up at the same place, but you see a lot of sort of order convergence at these higher levels. Right? So one example is this, the evolution of the ability to metabolize citrate in these strains. And if you look at this, you have 3 different strains that eventually evolved to metabolize citrate. They do it in different ways. There's some commonalities they share, but each pathway is going to be a little bit different. So if you're looking at the pathway again, there's contingency. If you zoom back and look at a bigger level, is there some type of order or structure that's driving the evolution of certain traits or evolution of this property? And that's what I'm going to be looking at. This is one more quote from Darwin, because I think he saw this, and this is in his letters where he was replying to someone who was arguing that evolution that you described is just chance. And he says, no, it's not. So that, am I really arguing that nothing about life's history could be predicted or might follow directly from the general laws of nature? He says, of course not. The question that we face is one of scale or level of focus. Life exhibits a structure obedient to physical principles, right? So life has to follow the chemistry and the underlying physics in a sense. So we do not live amidst the chaos of historical circumstance, unaffected by anything accessible to the scientific method as traditionally conceived. So he did see that there's some order and there's some regularity there that can be probed. Okay. So the second point I want to look at is what structure and order is required for evolution to operate. And I'll briefly go through this. and this was something that I think I find that as physicists tend to get this intuitively. It's almost not even worth mentioning. But a lot of times biologists don't recognize this. I think or don't appreciate this as much. Okay, so if you ask the question, why does evolution work? Okay, there's pictures of me over a span of 20-some years, right? And if you look at it, I have the same big, large nose in 20 years. My nose is relatively stable. I have a sort of a stable form. And so we exist in a universe that allows for stable, complex entities to exist. And that's absolutely critical, obviously, for evolution to work. So if there's a virus that is going to select against people that have large noses because it cultivates there and kills you. If my nose is continually getting larger and smaller day in and day out, natural selection is sort of a moving target. It's not going to be able to hit it. You're not going to get differential selection that you normally get. All right? So the question is, why do we exist in a world that, or in a universe that allows for stable, complex, ordered entities to exist? Well, we do because if you look at what allows a stable organism to exist, it's largely because you're made up of stable proteins. So proteins will fold. They have certain flexibility. They do unfold, so they're not indefinitely stable, but they have a high degree of stability that they maintain form and function. Okay? They don't transiently, in certain pathological conditions, they'll lose their form, but they don't spontaneously usually switch from one form to another, and so forth. And the reason that they have that is because they are made up of stable carbon atoms and nitrogen atoms, and which maintain that structure, which goes and why do you have stable atoms? You can say, well, because of the forces and the physical laws, the physical constants, and so forth, that we are in a universe that's amenable to biological evolution. Right? So there are certain necessary conditions that we talked about this morning, and that order is absolutely essential. So biological evolution is not something that's substrate independent. Right? It can't just operate on anything. There has to be a certain amount of structure and order in the chemistry and physics for evolution to work. Okay? And biological evolution does not create this. This is sort of, as I put it in quotes here, a gift to evolution. Okay. Well, you might say, so what? Okay, so you have some order there that's needed to get any stable, complex entities, but once you have them, they just sort of evolve and do whatever happens. You get all kinds of randomness, and they go in this direction or that direction. There's no order there. Okay. Well, I want to sort of look at the possibility that this order actually significantly shapes evolutionary trajectories, and particularly protein evolution. Okay. Now, one of the things that I think facts that impressed me the most as going through sort of biochemistry classes as an undergraduate was the fact that the amount of possible proteins that seem to exist, protein structures, is very small given sort of the space of protein sequence. So if you took the protein space of all 100 amino acid proteins that have 100 amino acids in, the number of possible sequence is 10 to the 130th. This vast array. As far as evolution is concerned, it's sort of an infinite number. You're never going to be able to search through all those. Okay? That being said, if you look at protein databases, right, and look and see how many folds do you have in these databases, it clusters around 1,000 unique folds. Right? So you have this sort of infinite space of sequences, all converging down into about 1,000 different naturally occurring folds. Now, the predictions of, again, that's just databases of proteins we know, right? But based on predictions, people argue that there's probably somewhere between, and people predicted 500, that was 20 years ago, and obviously we crossed that number, but usually ranges from 500 to 8,000. Still a relatively small number given the sort of possible sequences that exist out there. Okay? Well, why is this? Okay. Well, one of the main reasons is because of the nature of amino acids and how they interact. So if you take amino acids and string them together in peptide sequences, you end up with them collapsing into basically two structures. All right. alpha helices and beta sheets. Okay? And as you can see, the alpha helix here, here's the beta sheet. And if you look at the structure of the amino acid, what makes different amino acids different from each other is the R group here on the side chain that sticks off here. And the interesting thing about alpha helices and beta sheets is that the R groups play virtually no role in stabilizing the alpha helix or the beta sheet. It's all the backbone interactions, the backbone from the alpha carbon and the nitrogen and the carboxylic acid carbon on the amino acid chain. So the alpha helix, basically you have all the R groups sticking off, like you have a roll of toilet paper, all the R groups sticking off. The backbone is what holds this thing together. And then with these sheets, the R groups stick above or below the sheet. Right? And what this says is that it is not the specific amino acid necessarily that holds this thing together. It's the chemistry of the portion of the amino acid that's common to all amino acids. Right? And these are the sort of the two major stable structures that they form based on the angles of the bonds that are sort of sterically possible. Okay? Now, in addition to that, once you have alpha helices and beta sheets, there's only a certain number of ways in which you can fold these things together, in which you can arrange them. And a lot of them have to do not so much with the actual sequence of the amino acid, but with the number of amino acids that are in a loop, okay? And so if you have a beta sheet, whether it turns to the right or turns to the left, is largely determined by how many amino acids separate the alpha helix from the beta sheet, not the actual specific sequences. And if you have 3, it turns one way. If you have 2, it turns another way. So a lot of these things are sequence independent, right? So you have this huge sequence space, but a lot of the folding doesn't depend upon the exact sequence, okay? And so, Michael Denton has argued that there are organizational rules based on the physics and the chemistry that sort of allow proteins to only fold into, and he argues about 1,000 allowable higher order structures. It might be, higher than that, but it's probably at most an order of magnitude higher than that in terms of the number of allowable structures. Okay. Now, one of the things that leads to these factors that affect protein folding is the chemistry of the amino acids, right? And you might ask, well, are biological amino acids special in some respect, okay? Well, they tend to be easier to form given the chemical abundance of the universe. So you find many of the biological amino acids are able to form and have been found in meteors and so forth. So they've been able to form so abiotically. Okay? But there's nothing sort of special so much about these amino acids. Okay? So if you look at this this diagram up here, it's looking at a number of amino acids and putting them in a three-dimensional plane based on their size, their hydrophobicity, and their pKa, so the things that cause amino acid side chains to vary. The green ones represent the biological amino acids. And they have a certain amount of diversity there. Okay? And if you randomly sample and look for 20 amino acids, it's hard to find 20 random amino acids that have as much diversity. Right? You can find a few that are a little bit better, okay? But these the 20 that are there in green allow for the amount of sort of diversity that will give you those 1,000 1,000 different folds. So they're not really unique in their protein folding capacity. So we didn't have to evolutionarily get to these 20 amino acids. Right? You just have to come up with 20 amino acids that have sort of a range of hydrophobicities, a range of pKa's, and a range of sizes, and you're going to be able to get that structural complexity. So there's a sort of the interplay of contingency at one level. If you zoom in, well, if you ran evolution again, for example, you might not get the same 20 amino acids, but you would probably end up getting the same three-dimensional structures because of the complexity of the way amino acids in general interact. Okay? So So the last thing to sort of look at is, well, you only have 1,000 possible folds. Well, how populated is protein sequence space? Right? So, a lot of people if you intelligent design arguments say, well, there's no way to get a folded protein because it's the odds of against it because sequence space is filled with unfolded sequences. So is it sparsely inhabited by folded sequences over here, where if you just jump in somewhere randomly in sequence space, you're not going to have a folded protein, you're not going to have a functional protein, nothing that evolution can work on? Or does it look like this? Is it relatively dense? Now, if you jump in here, you have better odds of finding a folded sequence. Okay? Well, there is, again, because of the sequence independence, evidence that their sequence space is filled with a higher abundance of folded sequences than you might at first imagine. And estimates range from 10 to the second up to 10 to the 10th, 1 in 10 to the second and 1 to the 10 to the 10th actually fold. I mean, those are numbers or ranges in which evolution can work. And one of the main reasons is that what drives protein folding is largely not the actual amino acid sequence, but whether the amino acid is hydrophobic or polar. It's called the HP sequence. And you can actually design protein just based on the HP sequence, whether the sequencing of hydrophobic and polar amino acids, to get to fold into different structures. So if you're just looking at, well, how many different HP sequences do you have in a 100 amino acid chain? It drops from 10 to the 130th to 10 to the 30th. There's this huge drop in sort of possibilities there. And then on top of that, once you have a protein that folds, the amino acids on the surface are much more mutable. Right? So the ones at the core tend to have to be hydrophobic. If you mess with those, it probably tends to unfold, although there's some variability there. So this might be even a high estimate. Okay? So you get numbers that are much more amenable, just in theory. Okay? I'm going to skip this slide and just end with what is the empirical evidence? And there has been, in the last 10 years, a number of empirical studies trying to figure out if you just took a random sequence, what is the likelihood you get any type of folding? And so in this study, they took 80 amino acid random sequences, and right in the middle of it was a thrombin cleavage site. And so if this sort of string portion here, which was the random sequence, were able to fold into a stable structure, you're going to protect this from thrombin being able to degrade it. And so this is sort of their selection procedure. And they found that about 20% of them could prevent thrombin cleavage. Now, a lot of these might just be aggregates and they're not actually folding, but if you take some of them and use spectroscopy methods, they took a couple and found that you do get some alpha helical formation. So you're starting to get some folding here at a relatively high rate from random sequences. There's a couple other studies that look at this with sort of minimal amino acid libraries, rather than using 20 amino acids, using 5 or 12, and using random sequences and using different techniques. They're able to find that, yeah, you get soluble proteins, and you also can at significant levels, and you can get ones that show particularly some alpha helical content. So you do get folding in these sort of artificial settings where you're looking at smaller amino acid sets at significant amounts of the time. Okay? So in summary, I'd say, why does evolution via natural selection work at all? Well, it works because of the underlying chemical and physical order. And without that, the contingent events are not going to work. Right? And that is what is primary. The order is foundational. And it allows the contingencies of evolution to uncover sort of the ordered forms that exist. And this order significantly shapes evolutionary trajectories. It dictates, for example, in the case of proteins, the low number of robust folds that are going to exist out there. Not only that, but it helps dictate the abundance of these in sequence space, that they're not as rare as you might at first imagine, okay? And that hopefully a contingent evolutionary process can find those, even if they, the number of folded sequences are there in 10 to the minus 10th of all these possible sequences. So I'll stop there and I'll open up for questions.